Bismillahirrahmanirrahim. Assalamu alaikum students. The topic of the discussion is descriptive terms in embryology, part 2. As we have already discussed this topic up till zygote. Now, after zygote, there will be the division of the zygote, and the process under which division occurs, that is called cleavage. This is the series of mitotic cell divisions of the zygote that results in the formation of early embryonic cells, blastomeres. So, zygote will undergo series of mitotic cell divisions. Please listen carefully that these are the mitotic cell divisions, and these are series of mitotic cell division of the zygote that results in the formation of the blastomeres. Now, what will be the size of the structure of the zygote? It will remain unchanged because at each succeeding cleavage division, that the cells which are coming from the cells from where they are getting division, the newborn cells which are more in number, but these cells become smaller. So the succeeding cell of the blastomeres becomes smaller to accommodate themselves in the unchanged size of the zygote. So cleavage is the series of mitotic cell division of the zygote. Hence, first we have the one cell, then from the zygote we have the two blastomeres. Here you can see the zygote was fitting with this. So now two cells are fit in this structure, size remain unchanged, and after that succeeding cell division there will be the 4, and these blastomeres are smaller than these cells, and after the 4 they will get 8, and after the 8 we will have the 12 to 32 cells. These cells are called blastomeres, and you can check that the size of the structure is same, but as division occurs, cells become smaller and smaller to accommodate it within this structure. So when they become solid mass of 12 to approximately 32 blastomeres within that structure, then it is called morula. When there is the division of the zygote and the blastomeres are about 12 to approximately 32 in number, then the structure formed is called morula. First, the structure was zygote. We have the zygote. Then after the successive cell divisions, the structure is called, at the stage of 12 to 32 blastomeres, it is called morula. To become the morula, the blastomeres change their shape and tightly align themselves against each other to form a compact ball of cells. So this compact ball of cell is formed, that they will change their shape according to their position, and they will align themselves tightly with each other to fit in this structure. This change in shape and to tightly align themselves against each other This phenomena is known as the compaction. So, with the help of the compaction, that they change their shape and tightly align themselves against each other to form a compact ball of cells, and that is called morula when the number is from approximately from 12 to 32 blastomeres. The phenomenon compaction is probably mediated by cell surface adhesion glycoproteins. The morula stage occurs 3 to 4 days after fertilization, just as the early embryo enters the uterus. Then after morula, we will have the structure that is called blastocyst. Blastocyst. If you recall the previous, the cells are called blastomeres, and this structure is now called blastocyst. After 2 to 3 days, the morula enters the uterus from the uterine tube. Soon a fluid-filled cavity. Fluid-filled cavity. The blastocystic cavity develops inside it. This change converts the morula into a blastocyst. You can see this, that this is the morula which is present in a ball-like mass, and this is fluid-filled cavity which appears within this cell ball. So this is called blastocyst, so that the cells are present in its outer lining and cells are present inside. The cells which are present inside the circle, this is called inner cell mass, and the other name is the embryoblast. This is called embryoblast or inner cell mass, and this is called outer cell mass or the trophoblast. So this is called outer cell mass or the trophoblast, and this is called blastocystic cavity. This is called blastocystic cavity. So you can see that this is the structure in which the division occurs. This is the structure in which division occurs. This is called zona pellucida. Now, as this structure has get entry into the uterus, it will be embedded within the uterine wall. There will be the degeneration of this structure, zona pellucida. So here is, you can see it has been fully degenerated, and this structure is called blastocyst. This is outer cell mass. This is inner cell mass. Inner cell mass is also called embryoblast, so it will give rise to the embryo. Outer cell mass is called trophoblast. It will give rise to the placenta. The pole on which side embryoblast is present, that pole is called embryonic pole. The pole opposite to the embryonic pole is called the abembryonic pole. So blastocyst is formed when a fluid cavity develops inside the morula. This change converts the morula into a blastocyst. Its centrally located cells, the inner cell mass or embryoblast, is the embryonic part of the embryo. Then come to the implantation. I already told you that here zona pellucida is degenerating and here it has been fully degenerated. So implantation The process during which the blastocyst attaches to the endometrium. Here blastocyst attaches, zona pellucida, it has completely been degenerated. So the process during which the blastocyst attaches to the endometrium, the mucous membrane or lining of the uterus and subsequently embeds in its wall. Clear? Implantation is the embedding of the blastocyst into the uterine wall. The endometrium is the innermost layer of the uterus. So, the process during which the blastocyst attaches to the endometrium, the mucous membrane or lining of the uterus and subsequently embeds in it. Endometrium is the mucous membrane or lining of the inner lining of the uterus, and blastocyst attaches to it and subsequently embeds in it. And the day when it embeds in it, it is the 7th day. So the pre-implantation period of embryonic development is the time between fertilization and the beginning of implantation, a period of approximately 6 days. So pre-implantation period is the 6 days. Now coming to the gastrula. Gastrula. During gastrulation, transformation of a blastocyst into a gastrula, a 3-layered germ disc is formed, and it is during the 3rd week of development. Then the embryo or the conceptus is called the gastrula. 3 germ layers of the gastrula are ectoderm, mesoderm, and endoderm, and they will subsequently differentiate into the tissues and organs of the embryo. So this is about the gastrula, that 3 germ layers will be formed. After that, coming to the neurula, the early embryo during the 3rd and 4th week, when neural tube is developing, then this developing embryo is called neurula. The early embryo during the 3rd and 4th week, when the neural tube is developing from the neural plate, then this developing embryo is called neurula. It is the first appearance of the nervous system and the next stage after the gastrula. Now this is about the few of the terms in embryology. Now coming to the control of development. How the development occurs. Development occurs by these 4. Cell differentiation. They will be differentiated to form the different tissues or organs. Regulatory cell migration. That they are differentiated to the tissue which is away from their origin, so they will be migrated there where needed. Induction. So one type can induce the other type to form the different types of tissues. Apoptosis. Programmed cell type. Now we discuss this one by one. Cell differentiation. Cell differentiation is the process by which a less specialized cell become a more specialized cell type. Cell differentiation is the process by which a less specialized cell become more specialized cell type. Here, first cell type is the totipotent cells, a cell that is able to differentiate into all cell types including the placental tissues, for example zygote. From the zygote, we have the all cells types which can form all types of the tissues. Pluripotent cells. These cells have the ability to form all adult cell types, that is into any of the 3 germ layers. Totipotent was the cells that they can form all type of the cells including the placental tissues. Pluripotent cells that they have the ability to form adult cell types, not of the placental tissue. Now, coming to the multipotent cell types, they can give rise to other type of cells, but it is limited in its ability to differentiate. For example, hematopoietic stem cells, a blood stem cell that can develop into several types of blood cells, but cannot develop into brain cells or other types of cells. So they are now specific, more specific. So totipotent, that we can have all types of the cells, for example zygote. Pluripotent, that we have this ability to form the adult type of the cells, that is all three germ layers. Multipotent, it is more specialized, that we have the cell types which are limited in its ability to differentiate. For example, hematopoietic stem cells, they will produce all types of the blood cells. Now coming to the second point, that is regulated cell migration. Embryogenesis involves association, dissociation, and migration of cells. That is, first these cells are associated with each other. When we want these cells somewhere else, then they will show dissociation due to some molecular processes. Then dissociation will cause the migration of the cells. So regulated cell migration will be involved for the development. Then number 3 point was the induction. Induction is the interaction between the two separate histological tissues or primordia in the embryo that results in morphological differentiation. So one cell type will induce on the other cell type to result in morphological differentiation. So induction is the interaction between two separate histological tissues or primordia in the embryo that result in morphological differentiation. So we have the control of development by the cell differentiation, regulated cell migration, induction, and in the last we will discuss the apoptosis. This is the programmed cell death, important and normal component of the development of multicellular organisms. As you can see here, the development of the hand. Within the fingers, we have this tissue, and this tissue will be eliminated by the apoptosis, that is programmed cell death occur, and this web-like tissue will be eliminated. So this is all about the descriptive terms in embryology, and the control of development is also discussed in this lecture. Thank you. Allah Hafiz.